I am going to talk about a different type of experience in bringing complex data, and in particular genetic data, to the point of care. This will be from my perspective as the director of our Center for Personalized Medicine at the University of Colorado. When we think about personalized medicine, we think about many different types of information, and essentially bringing those together to create actionable insights that we can supply to healthcare workers and providers to be able to provide better care for individuals. And in this case, we're going to focus almost entirely at the combination of genes and medicines. So our center was created about 10 years ago to essentially both advance research, education, and care. If you want more kind of about the founding story, there's this American Journal of Human Genetics article that talks about kind of the original vision and kind of some early stage implementation. Today I'm going to focus almost exclusively on the implementation side, so how you actually bring this information back to the point of care and how you do this at scale in ways that support providers in making better decisions for individual patients. So very little on the research, very little on the healthcare worker side. Our implementation partner in this is University of Colorado Health, so UC Health. This is a little bit out of date now. It's 16 hospitals, more than 2,000 hospital beds, about 2.5 million unique patients per year. So just as you're thinking about context, and it covers the state of Colorado and then kind of reaches into some surrounding states. This is a story about how our process actually works for enrollment.
Your one-of-a-kind uniqueness could help improve the health of humankind? By participating in the biobank at the Colorado Center for Personalized Medicine, your genetic data and medical records could help advance healthcare for everyone. Medicine is not one size fits all. Sometimes individual patients need unique treatment. CCPM is researching how our genetic differences may help predict, diagnose, treat, and prevent disease. Researchers learn more about certain diseases by looking at the biobank's genetic data and medical records collected from thousands of people like you. With more participants in the biobank, they get a more comprehensive view of how genes influence health and disease, which could lead to huge innovations in personalized medicine. Participation is easy, and no extra appointments are needed. To join, patients can read and sign the consent form in their My Health Connection portal, giving us permission to look at information in your medical records. The next time your doctor orders a blood draw, additional samples will be collected for the biobank at no cost to you. That's it. Your samples will be linked with information from your medical records and could be used for a variety of studies, both by University of Colorado researchers and in partnership with companies developing treatments and diagnostic tests. Our patients' privacy is important to us. That's why all biobank research is protected, and your samples and medical data will be stored securely. If the biobank finds important information about your genes that may affect your risk of disease, response to medication, or disease carrier status, these results may be returned to your UC Health medical records. Not all diseases are tested for, and it may take several months or years before any results are returned to you, if at all. If you're ready to help humankind with a simple blood donation, join this important research effort by signing up today in your My Health Connection portal.
So important things about how we implement, we really try to automate as much as possible. So this is really streamlined into the patient experience. The consent, for example, is fully built out in the EHR. So when a patient goes to check in for their appointment, they're offered the opportunity to consent for the study or to decline to participate. If they consent, the next time they have a blood draw, another tube is drawn and used for the study. So it's really built out in this kind of eminently practical way, right? The goal is, with how people already interact with healthcare and with how healthcare systems already work, can we implement in that context? Through this, we now have more than 250,000 consented participants. We've got more than 170,000 samples. And so in terms of biobank studies, it's a relatively large study, both in terms of the number of participants and the number of samples. So we've got kind of three main areas for impact that were also kind of discussed in the video. I'll focus almost exclusively here on actually the implementation of personalized medicine. I added this slide while Paul was talking. This is one of the, I think these are the para papers that he kind of alluded to. This one is from just over a decade ago now, and it essentially argues that while we should think about return of results from biobank studies, this return of results is not feasible. It's going to cost too much, and thus studies shouldn't be required to do it. There's a complementary piece that was from a few months later that essentially argues that while yes, it is expensive to return results, it turns out that it's also the right thing to do. And so both of these things came out when CCPM was sort of being instantiated. I wasn't there at the time, but in my understanding is they were widely discussed. Our biobank was set up with the intent to return results. And so that has been kind of a guiding principle. So, at this point, what we like to say to people is, what we try to do is deploy genetics to move from sort of the standard treatment plan to an individual specific treatment plan. Most of what I'm going to talk about in terms of the examples here are going to be pharmacogenomics. And so often these are situations where an individual will metabolize a drug potentially differently than kind of the population average. So they might need more of the drug, they might need less of the drug, or they might need a different drug. And so that's what we're talking about, is really trying to optimize either dosing or medication selection given someone's kind of drug-gene interactions. And yeah. That's probably enough pharmacogenomics for this. If you want to talk in detail about pharmacogenomics, happy to chat more. Going to sort of how we design and implement, the way that pharmacogenomic testing is usually done is that a patient has an adverse reaction to some medication. For example, they're on a certain chemotherapy and they have a severe adverse effect. And so then a provider might say, well, why did that happen? Well, there's certain pharmacogenomic interactions that could lead to, for example, patients building up certain chemotherapies in their body and having these adverse effects. So if you're interested in why it happened, you can order pharmacogenomic testing. You can get a very long multi-page PDF that will tell you about all the variants that individual has, and you can decide to scan that to figure out what went wrong. You can imagine this usually doesn't happen, right? Most folks do not, in fact, order the pharmacogenomic test. So what we've focused on is really building out, ideally, a fully preemptive testing. So that means we test before any interaction is known, or even before a drug is ordered for which that test might be needed. And then the idea is, instead of telling people what went wrong, trying to get as far up in the process as you can, where you can tell providers exactly what they should do at the time they're making prescribing decisions. So we want that information to be there in advance of any of those decisions. And that's essentially how we've decided to build strategically all of our processes and workflows. So this is an example of a 58-year-old man who was receiving capecitabine for rectal cancer. In this case, the provider ordered Zofran for chemo-induced nausea and vomiting. Now, normally that would happen. The provider would order Zofran, that patient would be provided Zofran, and hopefully it would work for them. But one of the things that you could know if you had preemptive pharmacogenomic testing is perhaps this patient is actually an ultra-rapid metabolizer, so they have a variant in the gene CYP2D6 that makes them metabolize this drug very quickly. And so what that means is this drug is unlikely to work well for this patient, right? It's going to be cleared from their system before it can really have the therapeutic effect that we'd like it to have. So ideally, the physician would be informed as they make the prescribing decision, hey, this is probably not going to work that well for this person. We recommend changing them to another medication. So this is what we'd like to see. And this is, in fact, an actual patient story. One of the things to know is you might say, well, okay, that's great. So for that patient, it could make a difference. But for how many people is this going to make a difference? Like, how many people have a pharmacogenomic variant somewhere? Turns out almost everyone. They're essentially everywhere. So if we look just at the pharmacogenomic variants that we return, we have about 90% of our participants have at least one polymorphic PGX phenotype. So for at least one of these genes, the indication for the dosing recommendations or drug selection would potentially vary if they were provided that medication. And about half have two or more polymorphic PGX phenotypes. And then one of the things that's important to know is that there's literature that suggests that preemptive PGX testing can actually reduce the risk of adverse events by up to 30%. And so, this is something that we know can be effective, but the testing is not often done. But for most people, there's at least one polymorphic allele, and often two or more, that would have an influence here. And so that's the world that we want to enter, is we really want to provide this information up front to providers so they can make the right decision at the right time. As we built out CCPM, we've really designed for automation. I won't go through all of this, but we go all the way from running the tests in the biobank lab through supporting the construction of the clinical decision support that actually appears for providers. Throughout the entire process, we've designed for automation. So we want this to be as automatable as possible, as scalable as possible, and to Paul's point, as inexpensive as possible. So this is what Epic's Genomic Indicators tab looks like. If someone orders pharmacogenomic testing, they usually get a 90-page PDF. But if they happen to order testing from a provider that is plugged into their EHR so that it comes back as structured information, they get this really nice table, which again is still very cumbersome, but is in fact much better than your 90-page PDF. So what we do is we try to go a step beyond this. So while this information is available to our providers, so we do return results as structured information that ends up in the Genomic Indicators tab, we also have clinical decision support built around it. So this is the example for clopidogrel for a CYP2C19 intermediate metabolizer. So if a provider is going to order clopidogrel for one of these patients who's an intermediate metabolizer, instead of having to go to the Genomic Indicators tab every time they want to place an order and say, is this individual an intermediate metabolizer, we use clinical decision support to essentially, this is a severe alert, so we actually pop up an alert and say, hey, we recommend you remove this. We recommend choosing an alternative med, and here's why. And so this implementation means that while that Genomic Indicators tab is an order of magnitude better than what existed in the past, we can actually go beyond that and actually provide providers the information they need exactly when they need it. And this we find very good adoption with. I'll talk about that a little bit later. The other nice thing about this is you can scale this very quickly. And so in terms of genetic results returned, discrete genomic indicators returned to the electronic health record, we've grown a few thousand-fold in the last few years. Just for perspective, this was the end of June. So July 1, we had 925,000 discrete genomic results in the electronic health record. Next month, we'll cross 1 million. So when you're doing things as fast, growing 100,000 genomic indicators every month and a half or so, provides the opportunity to really get this back to patients and see that impact on healthcare. We can also ask, how, given this sort of information sitting in the electronic health record, how many patients have had at least one pharmacogenomic alert? 3 years ago, that was about 500 patients. Now it's just about 17,000 patients. So we can see these very large increases in scale in terms of the number of patients who are actually impacted by the potential pharmacogenomic findings. To dive in a little bit more into the actual results returned, so of the 900,000 results returned by the end of June, we had about 85,000 different participants who'd had a result returned, about 23,000 drug gene alerts in practice. And then our breakdown, it's actually on this side up here, can give you a sense of kind of what drug classes these are primarily impacting. One of the important things we always ask is, what's the actual impact on provider decisions? And so how many actual actions were taken? So we're now over 1,000 actions taken in response to the PGX alerts. We have a team that thinks very carefully about whether those alerts should be passive, like inline med warnings, or whether they should be active, so things that actually pop up in a clinician's workflow. We know a couple things. So the first thing is, if you pop something up in a clinician's workflow, they are more likely to respond to it. On the other hand, if you pop things up in a clinician's workflow all the time, they're more likely to begin to ignore it. And so we've designed this very carefully to ensure that the most severe drug-gene interactions are interruptive alerts. Everything else is a passive inline med warning. And what this means is for our highest risk pharmacogenomic alerts, we end up with very, very high compliance rates beyond what we see in the literature. But that's because the team behind this has thought very carefully about when you actually want to interrupt a physician's workflow and is trying to interrupt the physician's workflow only when it is absolutely essential. One of the nice things about having this biobank study is you can kind of quickly find an opportunity and then move it to making that implementation standard of care. So here's a story. This was a participant. This is not the actual picture of the participant. This is a stock photo. There was a participant at the end of 2022 where we had just gone live with our DPYD testing and our DPYD alerts. So they were seen for an oncology appointment in a community setting. 5-FU was prescribed for treatment. There's a DPYD 5-FU interaction that essentially leads to, for people with this genotype, an accumulation of this chemotherapy in their body to levels that become toxic. It can put them in the ER. It can also kill them. In this case, what happened is an alert fired that let the provider know, hey, this patient's an intermediate metabolizer. The oncologist proactively reduced the dose, and then even with that, they had to do a subsequent dose reduction. But without the initial alert, there's always a concern that that patient's going to end up potentially in the ER just due to toxic accumulations of 5-FU. And so we were able to take this to health system leadership and identify, hey, this potentially saved this patient from accumulating toxicities. And this could be a good opportunity to think about implementing this as standard of care. This predates, for example, the FDA labeling that is now is being implemented around this drug-gene interaction. So we've also pivoted so that we're not only running a biobank study. So everything I've told you about this far is on this side. It is a research study. Patients see that same video I showed you, and they have the opportunity to consent. If they consent, we take that extra sample. We run it in the lab and return results to the EHR, but that's essentially population-based, and it's not like we're trying to identify high-risk populations and just intervene with them. It's all around kind of a research consent. They participate in a research study. What I'm going to shift gears to now is some work that we've done in GI oncology. So this is clinical first, so there is no research consent. This is making genetic testing in GI oncology standard of care. And so what we've done there is essentially taken the same ideas behind the process that we use for the biobank study, but using this DPYD patient as an example, and then built this into the routine clinical workflows, initially at our academic medical center. And now, as of August 4th, we'll be live across the entire UC Health system. So this was what we did. So we sort of said, okay, we have this one patient that we've seen through the biobank that sort of leads to this individual story. Could we envision what the patient impact would be like if we did this type of testing in every GI oncology setting? So we imagine that there's a patient seen at the GI oncology clinic. Their oncologist gets a clinical decision support alert that says, hey, order this test. Ideally, we would return those first results within a week because we know they're likely to start chemo in a week. Or have surgery and then start chemo in a week. So we want to get those results back within that first week window. You can imagine we return those initial results, which would focus on things that might impact chemotherapy decisions. And then subsequently, we also know that individuals who are undergoing cancer treatment will also receive a lot of treatment with supportive care meds. And so we would ideally also return information that, like, for example, the Zofran example that would impact the selection of either the selection or dosing of supportive care meds, so things that people often encounter when they're going through cancer treatment. So that patient was in December 2022. We got a really fast green light on this, and actually, because we have so much of this process in place, we're able to implement it successfully in less than a year. So those of you who know about technology products in technology implementation in healthcare systems know that most things take 24 months and more than half of deployments fail. So we were happy to deploy in 8 months and successfully. Over the first couple years, we've had now 529 patients who've actually had results go all the way through kind of initial blood draw through return of results. I'll go a little bit into what we return. So we return, yeah.
So how do you decide you have enough evidence to take action? Because genomics is like there are a few cases where we have large effects, but then a long tail of unclear whether we should.
Yeah.
That's the question.
Yeah. So the question is, that's all good. So the question is, how do we decide what has reached the stage where it actually makes sense to implement? So we have, first we use the CPIC-PGRN guidelines as kind of a starting point, and then we have a team that essentially goes through the guidelines and recommendations and evaluates the strength of evidence as well as the potential severity, and also how implementable things are. So there's stuff, for example, that we cannot readily implement on the genotyping strategy that we use, but that might be useful. So all those things are considered. But yeah, I agree. If you're going to provide clinical decision support, the bar for evidence has to be very high. It can't be some kind of suggestive potential interact. Yeah, like a GWAS result is not going to be sufficient. Like you need to understand the mechanism by which this variant actually exerts the effect. Yeah. This is live now. As of August 4th, it'll be live across the entire UC Health system. So essentially all GI medical oncology patients, actually this says at Metro Clinics because I didn't update this slide, but GI medical oncology patients across the entire system who are going to receive 5-FU, capecitabine, or irinotecan. We do preemptive PGX testing for all of that. And as well as a set of supportive care meds, which I want to reach to this side, because on my laptop it looks like they're on this side, but they're over here. Let's see, I think I didn't put the timeline in here, but we get the results back for the chemo-related findings in about 4.7 days, so faster than essentially any commercial lab that I am aware of. And then for the supportive care meds, we actually get the results back within 17 days. And we know that that kind of 7-day mark is a key mark for kind of when chemo might initially be prescribed. And then 21 days was our target for the supportive care meds. So we're able to get this back very quickly because it builds on this kind of backbone that we've established through the biobank research study. Just to get a sense of the impact, about just under 10% of patients actually have one or more high-impact chemo PGX alerts that actually end up firing, there's a lot of drugs that are used in cancer treatment, not terribly surprising, especially supportive care meds. And so about a third of patients actually have a supportive care drug-gene interaction alert fire. So, and we actually have a team of pharmacists who actually go through each of the high-impact chemo alerts and make sure that the providers are actually paying attention to the results that they need to pay attention to when they're making care decisions, because those are high risk, and we want to make sure that that the right decision is made. So finally, I just want to talk just a little bit about results that go beyond pharmacogenomics. So Paul mentioned we call these high-impact pathogenic variants. You can look at 10 different institutions. They'll probably have 10 different names for these. What these are essentially things like BRCA or other kind of relatively high impact genes that predispose one to certain conditions. We focus only on those that are actionable. So for example, we would not return something like a Huntington's disease gene because it is not actionable. There's no kind of action the individual can take to prevent that. We would return things like BRCA status because there are healthcare decisions that an individual can make to, based on understanding their genotype there. So we've also grown this program over the last few years from tens of results returned to hundreds of results returned. We'll probably cross 1,000 results returned over the next few months. Just to give you a sense of what we see, this is kind of a deep dive. We've got more than 1,000 results that could be returned. Many of them there's a few different things that happen. So for these, we have a genetic counselor that actually reaches out to folks and first discusses the potential to return results, the potential impact, and what that any actions that person might want to take just generally, not with respect to the specific result. The individual then can consent to have that result returned. If they do consent to have that result returned, then there's a follow-up call where that actually, where that information is shared with them. Just to give a sense, there's a fair number of people who never get back to us, which is probably not terribly surprising. Some people withdraw, so if you're about to tell them something, they decide they do not want to hear it, they would rather withdraw from the study. And then there's a set of other potential results, but about 60% of the time we actually have gotten the result returned to individuals. Just to give a sense of why people withdraw, so here's some examples. Sometimes they just don't want to know, there's a lot going on in their life. Sometimes they actually have the information, it's just not documented in their medical record. So I should say a fair number of these, there's actually information in their medical record that shows the participant's already aware. It's just, but they have genetic testing before we would provide it. There's a number of other reasons that potentially folks either withdraw or are withdrawn before a result can be returned. For those who have had results returned, the majority are either cancer or cardiac conditions. So we heard a lot from Paul about BRCA status. That is, those types of cancer predisposition genes are the largest. They're a plurality of the results that we end up returning. To one of Paul's points, what do people do with the information afterward? This is what we've seen. So obviously it's going to be different in different settings. But in our study, about 70% of the people will actually review the result with their PCP. 30% with a specialist. More than half are actually getting some test or follow-up as a result of the genetic result that we report to them. One of the things we are grateful for is that most folks report having no problems getting the medical tests that they elect to pursue completed. There are a subset that do have challenges. I know some of the questions before around insurance came up. And so we do have folks who have had results returned who have reported those challenges. One of the things that we were happy to see is that 85%, so the vast majority of people, do not report having problems paying for care related to the result. Because one of the things you want to be careful of is if you're going to return a genetic result back to someone that is nominally actionable, if the healthcare system and the way that we pay for healthcare in this country does not allow those individuals to take that action, that is, instead of being empowering, that can be you can be putting a challenge in someone's life that they cannot deal with. And so we are happy to see that in many cases, in a majority of cases, people do not report problems paying for care as a result of the information that we're returning to them. I have one more video. This is from one of our participants who just was excited to get their result returned and then ended up talking to a news station. So I'll give you a sense of what at least some of our folks have to say.
All new here at 9, a local woman would have never known that she had a potentially deadly condition without the help of some new technology.
And she submitted her blood to the biobank at UC Health, just hoping to help researchers, and never imagined they'd make a discovery that changed her life. Fox 31's Samantha Spitz joining us in studio with the story you'll only see right here on Fox 31. Samantha.
Yeah, pretty incredible what technology can do nowadays. Pam Dyer submitted her blood to the biobank at UC Health after getting a routine blood draw from her doctor. The results came. It's quite a shock.
Yes, silly.
Sitting still is not in Pam Dyer's vocabulary.
I've always been active my whole life. Runner, hiker, whatever Colorado does, I do it.
But after submitting her blood to the biobank at UC Health following a routine blood draw with her doctor,
it was a shock when I got the call.
Pam found out she had a heart condition that, if left untreated, could have led to heart failure.
Never in my wildest dreams would I realize that just saying yes, checking a box, and having my blood contributed to this big pool of population of DNAs, that it would have a direct correlation to me, and I would benefit hugely.
So far, we've returned over 350 results to patients like Pam.
Christy Crooks says patients can opt to send their blood to the biobank for free, where it's used for research and new studies. And at the same time, alert people if they're at risk for developing a certain disease or condition.
What we're hoping to do is empower patients and their providers to access care earlier and prevent the really severe outcomes. Like with Pam, having access to this cutting-edge drug through UC Health means that her progression is likely to be less than it otherwise would have.
She says researchers also study how genes can influence response to medication, which can help patients and their providers choose the best medication in certain circumstances.
I'm just so fortunate. It's kind of like a win-win. They found it, it's there, but there's something that prevents it. So I can just keep on living and doing, what I want to do without any impacts from the disease that I'm carrying.
Now, the biobank at the Colorado Center for Personalized Medicine is not target genetic testing, but rather a research lab that also returns crucial genetic results to participants who have consented to have their DNA studied.
All new here.
Okay, so with that, I'm going to kind of close the chapter on talking about how we get results back to patients. Love to take questions on that. I will just very briefly talk about how we support researchers on the campus and also provide healthcare workers with kind of unique opportunities to sit at the cutting edge of care. So, one of the things Paul also mentioned is kind of the power of these types of resources for research. So if you are, for example, a University of Colorado researcher, you have access to this resource that draws, actually, I think, from all participants from all 50 states, but mostly in Colorado. This includes, and for the entire set of participants, we've tied their genetic data back to the electronic health records, so we can provide information like Phe codes if you want to do PheWAS or other types of studies across this entire cohort. There's a lot of different ways that folks use this. Some ways that you'd imagine up front, other ways that you might not. So there's a lot of population geneticists who use this to study a set of different conditions. We also provide the DNA specimens to researchers at Colorado with certain questions that require access to the underlying genetic material itself. And then finally, some of the interesting things that folks do is end up, for example, recruiting participants based on a certain genotype for studies. This can be useful in clinical trials. It can also be useful if you want to study certain gene-environment interactions. We have now, with UC Health built out through the Recruit tool, essentially a way to take that electronic consent process that I talked about and essentially slice individuals so that you can take, like, okay, I want to do an electronic consent for my study, but only of people with this genotype, for example. So that whole thing can be rolled out. There's the ability to use, through this, essentially patient portal messages to be able to proactively reach out to individuals who've indicated an interest in being included in research studies. So there's lots of interesting stuff that you can do to advance research using the genetic data that comes from these types of research studies. And then finally, I'll just briefly mention the healthcare worker experience. So we have a bunch of different ways that we think about empowering healthcare workers. That includes a personalized medicine certificate program. That's often taken by folks at our institution, but it is an online-only program. So there are people who, from around the country who do elect to take it. There's a build-out for a master's program. We have a lot of engagement at medical executive councils. The thing I'm actually going to talk about is, very briefly, is the Biobank Champions program. So this is a program that's designed to engage frontline healthcare workers. So these folks essentially decide that they are a busy healthcare worker, and so what they would like to do is take on something additional above and beyond that incredibly stressful job they already do. They decide that they basically say, for the next 2 years, I want to participate in this Biobank Champions program. I'm going to learn more about the biobank. I'm going to be essentially the subject matter expert for the biobank within my unit. They tend to be phlebotomists or other technicians. So here's a few examples. One of the things that we like to see is that this type of program, instead of increasing burnout, increases healthcare workers' satisfaction and retention. So that is one of the things that we end up seeing. These folks end up very enthusiastic about their role in the biobank and sitting at the forefront of healthcare. So at a time when many medical executives are struggling with how to keep their workers engaged and enthusiastic about the mission, I think this is a good way for hospital executives to essentially see a win-win. Their staff end up happier and more enthusiastic, and their patients get better care. I won't read all those because hopefully you got a chance to glance at what they have to say. One of the other things that's nice, in addition to just improving the understanding of genetic medicine that healthcare workers across the enterprise have, we also see that it's actually improved the biobank itself. So for units that have onboarded a champion, we see an 86% increase in biobank samples collected. So one of the things I said is that this process is fully automated. It is a fully automated process, except the final step of the blood draw, right? And so for that blood draw to happen, even if the patient's consented and indicated they want that to occur, the healthcare worker has to provide that opportunity to them. And so we see that units with these champions have a much higher increase in actual collections that align with the number of consented folks that are seen in those clinics. And then, you can see some more information. The CE credits, I think, are a big win for the champions as well. So with that, I want to have time to answer some questions. So I think we have like 5 to 7 minutes left. I just want to say thank you to the entire team. So both within CCPM and then the teams we work with at UC Health and the CU Anschutz School of Medicine, and essentially everyone involved. It is a very large project with many people involved, and I owe a debt of gratitude to everyone who makes this possible. And with that, I would be happy to take any questions except about getting people to pay for this.